Good morning, and welcome to the third of three videos on the first section of Chapter 2 in your textbooks entitled The Neuron. We'll start this video with a brief discussion of brain scans. Now, you may ask why we're talking about this right now, but the answer is very straightforward. When you're doing research, whether it's biological or psychological, there are times when you need to know what part of the brain is actually in charge of whatever it is that you're trying to examine. Right, movement of fingers, or thought processes, or things that control sleep cycles, or speech, or hearing. Right, and in order to do that, you need to be able to scan the brain and to know what areas of the brain are actually in charge of those sections. The two most common types of brain scans are what we call PET scans and fMRIs. Now, a PET scan, PET, stands for positron emission tomography. And the way that this works is as follows. All right, you introduce a radioactive sugar into the bloodstream. Now, those of you who remember from your high school biology, the main fuel of a cell is sugar, specifically glucose. All right, so when you introduce this radioactive sugar into the bloodstream, the area of the brain that is active is going to use more sugar than areas of the brain that are not active. And this metabolized sugar then releases proteins, positrons, not proteins, positrons. All right. The positrons are then detected and measured. So positron emission tomography, PET scans. All right. And some of you may have had a PET scan. All right. And fMRI can be used on other parts of the body as well, not just on the brain. But the way that this works is that it detects the amount of blood flow into an area of the brain. All right. And because there's more blood flow to an area indicates that that area is more active than an area that has less blood flow. All right. So PET scans and fMRIs are the two major types of brain scans that are currently used in order to examine working brains. All right. Now, we'll conclude this series of videos and our discussion of the first section of Chapter 2 with a talk about neurotransmitters, drugs, and poisons. Okay, just so that you are aware, we use the term neurotransmitter, but you have to be aware that there's over 100 different kinds of chemicals that can function as a neurotransmitter in the human body. Okay. And there are other chemicals out there that can actually influence how well those neurotransmitters do their job. Now, those other chemicals fall into two major categories. The first are what we call agonists. Now, an agonist is a drug or a poison that increases the activity of one or more neurotransmitters. All right, and so it pumps it up, it makes it work really, really well. An antagonist, all right, and you may know this term from, for example, English literature, right? An antagonist is the bad guy in the book or the movie, right? The agonist is the good guy. But an agonist really just means that it builds up the activity, and an antagonist is a drug or a poison that decreases the activity of one or more neurotransmitters. All right, so agonists and antagonists. All right, keep those two terms in mind as we go through some of these different neurotransmitters. Now, I'm not going to go through all 100 of these, but we'll talk about the most common 4 or 5 of them so that you have at least some idea of how they work. So the first one is acetylcholine. All right, and you saw this one earlier. It was abbreviated as ACH. All right, acetylcholine. And this is a neurotransmitter that's involved in learning, memory, and muscle movement. So you can see that this is obviously a very important neurotransmitter for our purposes here. Just as a an example of things that might go wrong if the acetylcholine levels aren't quite right. Alzheimer's patients, for whatever reason, have very low levels of acetylcholine. Now, whether this is what causes the low levels, and whether this is a cause of Alzheimer's or the result of Alzheimer's, is still to be determined, as far as I know. Botulism. This is essentially food poisoning. All right, but there's a toxin, a poison from food poisoning that causes your chest and diaphragm muscles that help you breathe to be paralyzed. And so the way that this works is that the botulism toxin is actually an antagonist to acetylcholine. So it blocks the release of acetylcholine. It doesn't let it get out of the presynaptic membrane. And therefore, the next nerve can't be stimulated. And so you can't move your chest muscles and your diaphragm in order to help you breathe. There's a poison called curare, which is from the poison arrow frogs that you find in South America. These paralyze your muscles, all right? And in this case, again, this is an antagonist, but the way that this one works is the acetylcholine is released into the synaptic gap, but the receptor sites on the postsynaptic membrane are blocked, and therefore the acetylcholine can't stimulate a signal in the next neuron, the postsynaptic neuron. And last but not least, black widow spider venom is an agonist. And so what it does is it releases all of the acetylcholine all at once, right out, okay, into the synaptic gap. And what that does is it stimulates the next set of nerves. It overstimulates them. And so initially it causes convulsions because the stimulation is occurring fast and furious. But once you run out of acetylcholine, it then leads to paralysis because you can no longer stimulate those muscles to move. All right. And so, again, those are all methods or ways that these things can interfere with the work of acetylcholine. Now, another neurotransmitter that you may have heard of is dopamine. All right, this is a neurotransmitter that's involved in arousal and mood states. All right, meaning waking you up and keeping you happy. It's important in thought processes and in physical movement. Things that can go wrong with this include things like Parkinson's disease. Parkinson's patients have difficulty moving things. All right, they have muscle tremors. If you've known somebody with Parkinson's, their muscles, they start moving and they can't stop. They kind of shake and tremor. They have difficulty starting movements, and their movements tend to be very rigid. And this is because of a lack of dopamine in certain nerves in the brain. I have no idea who that was. Don't care. Now, in any event, people with Parkinson's essentially can start a movement, but then can't stop it. The movement just continues. Now, addictive stimulants like cocaine, amphetamines, and things like that are agonists for dopamine. They deplete it in the axon terminals, and they prevent its reuptake, meaning they can't recycle it. And so amphetamines, for example, stimulate dopamine's continuous release, depleting it from the axon terminals. So it kind of hypes you up. It gets you overactive. Okay, cocaine blocks the reuptake of dopamine, and again, so this will keep you very, very active. All right, hyped up. Okay, very high, if you will. Okay, serotonin and norepinephrine. These are neurotransmitters that are involved again in arousal, mood states, happy or sad or that kind of thing, sleeping and eating. All right, and so feelings of well-being, happiness, and that sort of thing. And so if you don't have um, a good feeling of well-being and you're not happy, you may be depressed. And in this case, you might be prescribed drugs that will actually affect your serotonin and norepinephrine levels. For example, two examples, there are SSRIs and SSNRIs. An SSRI is a selective serotonin reuptake inhibitor. Okay, and these are antidepressants, and what they do is they block the reuptake of serotonin, so that the serotonin is still in the synaptic gap and stimulating the next neuron. And this again helps increase feelings of happiness and well-being. Examples of this that are still on the market are Prozac, Paxil, and Zoloft. You may have seen commercials for these. You may know someone who's taking one of these kinds of medications as a mood enhancer or an antidepressant. SSNRIs are selective serotonin and norepinephrine reuptake inhibitors, and these work in much the same way as SSRIs. Commercial names for these that are currently on the market include things like Cymbalta, Pristiq, and Effexor. Okay, just so that you know. GABA and glutamate are the main inhibitory and excitatory neurotransmitters in the central nervous system. GABA works to keep the brain from becoming too excited, too aroused, too hyper. Okay, and so tranquilizers like Valium are agonists for GABA. They encourage the work that GABA does, all right, and they allow it to function more properly, and they bring the mental state from a very excited state down to a more level state, if you will. Glutamate, on the other hand, is a neurotransmitter that's involved in memory storage and pain perception. And so these are typically used for lowering your ability to, well, I'm sorry, let me start that again. Glutamate allows you to detect pain, and it's involved in memory storage. And psychosis sometimes is triggering memories that are either false or overblown. And so antipsychotic drugs actually bring the level of glutamate down in the body. Last but not least, we have the endorphins, and these are involved in pain relief and feelings of pleasure. When they're released in the body, we feel less pain, and we feel a sense of euphoria. All right, you get that runner's high. All right, if you're an athlete and you work out, you run a long distance or that sort of thing, you are responding to the release of endorphins. It makes you feel better, makes you feel good. And the way that it does this is it triggers the reward center in the brain and actually causes the release of dopamine, all right, which again increases your positive feelings. All right. And so those are the various kinds of neurotransmitters. All right, that's only what 5 or 6 of them that we talked about maybe. And remember, there are hundreds out there. But this just gives you kind of an idea of how some of them might actually work. And again, all of these are involved in the gap between two different neurons. So anyway, so that brings us to the end of our videos on the first section of chapter 2. And I hope you learned something.